Welcome to Health Digest, your daily guide to healthy living. 5 food that kills and prevent breast cancer. In the ongoing global effort to combat breast cancer, our focus is rightly placed on advanced screening, genetic counseling, and cutting-edge treatments. However, a growing and compelling body of scientific evidence points to a powerful yet accessible adjunct to these measures: our diet. Today, we will delve into the world of nutritional oncology, specifically exploring 5 fruits whose biochemical properties have demonstrated significant potential in the prevention and supportive management of breast cancer. This discussion is grounded in preclinical and clinical research, focusing on how specific bioactive compounds can influence carcinogenesis at a molecular level. It is imperative to begin with a clear, professional disclaimer. The information presented is for educational purposes and is not a substitute for medical advice from a qualified oncologist or physician. These fruits should be viewed as components of a preventive, health-supportive diet and, for those undergoing treatment, as potential complementary therapies to be discussed with your healthcare team. They are not standalone cures. Our exploration will detail the mechanisms of action, the how and why, behind each fruit's potential efficacy. We will move beyond general antioxidant claims to specific pathways like apoptosis induction, angiogenesis inhibition, and estrogen receptor modulation. Let's get started. Our first category encompasses a family of fruits renowned for their vibrant colors and potent phytochemical profiles: berries, specifically blueberries, raspberries, blackberries, and strawberries. The primary anti-cancer agents in berries are a class of polyphenols called anthocyanins, which provide their deep purples, blues, and reds, and ellagic acid, found particularly in raspberries and strawberries. Research, including studies published in the Journal of Agricultural and Food Chemistry and Cancer Research, has illuminated their multifaceted approach against breast cancer cells. First, they are powerful anti-angiogenic agents. Tumors, to grow beyond a minuscule size, must create their own blood supply, a process called angiogenesis. Compounds in berries have been shown to inhibit the production of vascular endothelial growth factor, VEGF, effectively starving the tumor of the nutrients and oxygen it needs to expand. Second, and perhaps more profoundly, they influence apoptosis and cell cycle arrest. Ellagic acid and anthocyanins can activate enzymes that trigger programmed cell death in malignant cells while leaving healthy cells unharmed. They achieve this by modulating key signaling pathways, such as the PI3K ACT pathway, which is often dysregulated in breast cancer. Furthermore, berries exhibit epigenetic effects. This means they can influence how genes are expressed without altering the DNA sequence itself. Certain compounds in berries can help reactivate tumor suppressor genes that may have been silenced in cancerous cells. To maximize benefits of berries, opt for a variety of berries to get a broad spectrum of compounds. Fresh or frozen are equally effective, as the freezing process can preserve these delicate phytochemicals. Consider integrating a serving of mixed berries into your daily diet through smoothies, oatmeal, or as a standalone snack. The second fruit group on our list is citrus. Oranges, lemons, limes, and grapefruits. While their vitamin C content offers general antioxidant support, the true anti-cancer prowess of citrus lies in a less famous class of compounds, limonoids. Found in the pulp and especially the white pith, limonoids are responsible for the slight bitterness in citrus. The most studied limonoid, limonin, and its precursor, limonin glucoside, have shown remarkable activity in laboratory models of breast cancer. A pivotal study from the University of Arizona Cancer Center demonstrated that limonoids can induce the activity of a critical detoxification enzyme in the liver, known as glutathione S-transferase, GST. This enzyme helps the body excrete potential carcinogens, thereby offering a preventive effect. More directly, limonoids have been shown to inhibit proliferation and induce apoptosis in estrogen receptor negative, ER-negative, breast cancer cells. This is particularly significant because ER breast cancers are often more aggressive and have fewer targeted treatment options. They appear to achieve this by inhibiting cell cycle progression and promoting the expression of pro-apoptotic proteins. Additionally, Citrus flavonoids like naringenin from grapefruit and hesperidin from oranges have demonstrated the ability to inhibit the invasive capacity of breast cancer cells, potentially reducing metastasis. A critical note of caution: grapefruit and its juice can interfere with the metabolism of a wide array of pharmaceuticals, including certain chemotherapies and hormone therapies, by inhibiting the cytochrome P450 3A4 enzyme system. If you are undergoing active treatment, it is absolutely essential to consult your oncologist before consuming grapefruit products. Always choose whole citrus segments over juice to benefit from the fiber and the pith. Use zest from organic lemons and oranges in dressings and cooking, as it is concentrated with bioactive compounds. At number 3, we have stone fruits: peaches, plums, and nectarines. Recent research from Texas A&M University, published in the Journal of Agricultural and Food Chemistry, has catapulted these common fruits to the forefront of nutritional oncology. The research team found that extracts from specific varieties of peaches and plums were selectively toxic to even the most aggressive types of breast cancer cells, while leaving normal, healthy cells completely unaffected. The potency of the stone fruit extracts was comparable to that of the chemotherapeutic drug cisplatin, but without the harmful side effects. The active components are a combination of phenolic acids and flavonoids, including chlorogenic and neochlorogenic acid. The compounds in stone fruits appear to disrupt this aberrant energy production pathway, creating a state of metabolic crisis within the cancer cell. This crisis then triggers the mitochondria of the cancer cell to initiate apoptosis. The study particularly highlighted efficacy against triple-negative breast cancer cells, a subtype that lacks estrogen, progesterone, and HER2 receptors, making it difficult to treat with targeted therapies. When taking these fruits, choose ripe, deeply colored fruits, as the concentration of these beneficial phenolates is often higher. Both fresh and frozen varieties are excellent. When in season, make them a regular part of your fruit intake. Our fourth fruit is the grape, with a special focus on the red and purple varieties. The hero molecule here is resveratrol, a stilbenoid compound found predominantly in the skin of red grapes. Resveratrol has been extensively studied for its cardioprotective effects, but its anti-cancer properties are equally impressive. Its primary mechanism in breast cancer prevention and control is the potent inhibition of aromatase. Aromatase is an enzyme that converts androgens into estrogen in the body. In postmenopausal women, this is the primary source of estrogen, and high estrogen levels are a known risk factor for estrogen receptor-positive ER+ breast cancer. By inhibiting aromatase, resveratrol can effectively lower estrogen production in the body, acting in a manner similar to a class of drugs called aromatase inhibitors, though in a dietary, non-pharmaceutical capacity. Red grapes are the best source. As resveratrol is in the skin, eating them whole is crucial. While red wine contains resveratrol, the potential risks of alcohol consumption, a known carcinogen, outweigh the benefits for cancer prevention. Therefore, whole fruit is the unequivocally recommended delivery method. The fifth and final fruit on our list is the ancient and potent pomegranate. Its complex biochemical profile, containing L-gitannins, anthocyanins, and unique fatty acids, makes it a formidable opponent to breast cancer development. This enzyme is responsible for converting the weaker form of estrogen, estrone, into the much more potent and carcinogenic estradiol. By blocking this conversion, pomegranate compounds can locally reduce the levels of strong estrogen in breast tissue, thereby reducing its proliferative effect on ER+ cancer cells. Pomegranate extract has also been shown to significantly reduce the proliferation of breast cancer cells, both ER+ and ER-. By modulating the NFKB and MAPK signaling pathways, these pathways are like central switchboards for cell growth and survival. By disrupting their pro-cancer signals, pomegranate can slow down or halt tumor progression. Studies have also highlighted its synergistic potential. When combined with the anti-estrogen drug tamoxifen, pomegranate extract enhances the drug's cytotoxic effects on cancer cells, suggesting a supportive role in combination therapy. The whole arils, the jewel-like seeds, are the most beneficial as they contain the fiber, juice, and seed. The seed itself contains punicic acid, a type of omega-5 fatty acid with demonstrated anti-inflammatory effects. Pomegranate juice, particularly 100% pure with no added sugars, is also a potent source of these compounds. In conclusion, the scientific evidence for incorporating these 5 fruits, berries, citrus, stone fruits, grapes, and pomegranate, into a breast-healthy diet is robust and growing. Their power lies not in isolation, but in their synergy. A diverse intake provides a multi-targeted, broad-spectrum approach to cellular defense, impacting everything from estrogen metabolism and angiogenesis to apoptosis induction. Empowerment in health comes from knowledge and action. Let this information serve as a foundation for informed dietary choices that support your long-term well-being. Thank you for your time and attention. If you found this detailed analysis valuable, please engage with this content and subscribe for more science-based health information. Until next time, be well.